Thank you so much, sir. I will read on that.
Sure, sure.
Yeah, Ria, well presented. And I just wanted to let you know, you also go through the different types of primers. Okay, there are different types of primers which are available. I do not know about this particular paper. Do you have an idea of what kind of primers were used in this particular experiment or the paper as such? Have you read through?
Yes, ma'am. Nothing for primer as such was mentioned. The
Okay.
main focus of the paper was on enzyme. That's why.
Okay, fine. Right. Anyway, so an additional reading can always enhance your knowledge. Okay. Yeah. Thank you.
Thank you, ma'am.
Judges, I would like to ask you, should we take a screenshot of all the students right now? Because as and when they finish, they can leave the meeting.
Yes,
if
yes,
permit.
ma'am.
Okay, so
Yes, yes.
students, can you please start your videos? We can take a screenshot for the record, and then you can again switch them off.
Yeah, please excuse me. My camera is not working.
That is okay.
Yeah.
Sir, your photograph is good.
Yeah, yeah.
Students? Guilty. Just a minute, I'll click one more. Just tell me your. Yes, we can all switch up the videos now. Okay, so the next student. Is Chaitanya Katkal from SY Biotech. I will make you the co-host. Oh, you can just tell me your roll number. Yes, Chaitanya, you can start. Madam, any more questions?
Yes, ma'am. Just is my screen visible?
Yes.
Thank you, sir.
From TY.
Yeah. Yes.
After that,
Yeah. So a very good afternoon to respected judges and everyone present here. Myself, Chaitanya Gatkar, and I am pursuing a bachelor's degree in biotechnology and currently in second year. Today, I will be presenting the classical research paper, simultaneous amplification and detection of specific DNA sequence. So before diving straight into the paper, I would like to give a short overview about PCR itself and its principles. The word polymerase chain reaction is self-explanatory, as the word polymerase de- denotes the enzyme used, and chain reaction states that it repeats itself again and again. We all know that DNA such RNA is the genetic material in all living organisms, encoding genes and information important for survival in everyday life. DNA is primarily composed of a ribose sugar, nitrogen base, and phosphate group. The property of our interest is the formation of the phosphodiester bond, which acts as a backbone of DNA, linking a nucleotide to another. This property is utilized by the enzyme polymerase, which adds nucleotides to the 3' end of the DNA strand, resulting in an elongation of DNA in a 5' to 3' direction. Polymerase chain reaction uses this principle to make millions of copies of DNA. PCR consists of the following three processes. Denaturation. It involves heating of the DNA template to separate the double-stranded DNA into single-stranded DNA by breaking the hydrogen bonds between the complementary bases. Second, annealing. This step allows the hybridization of the template DNA with primer, as the primer provides a starting point of the DNA synthesis, since DNA polymerase can only attach nucleotides to an existing strand. This step is very crucial as the fate of the amplified product depends on it. Third, extension. In this step, DNA polymerase synthesizes a new strand complementary to the template. A PCR reaction mixture composed of the following components. First, the template. Second, polymerase enzyme. The buffer, primers, and the nucleotides, which are the DNA building blocks itself. Now, I would like to speak about the amplification curve, since the study of amplification curve is very important in detection of the viral load. Infamously, in case of the SARS-CoV-2 infection, it is created, the amplification curve is created when fluorescent signal comes from the sample. This signal is plotted against the cycle number. This forms a sigmoid curve in nature, as we can clearly see in this picture. This curve consists of following three phases. For the initial phase, this phase represents the signal received at the starting 3 to 5 cycles of the DNA. It is the minimum fluorescence detected and is also accompanied by background noises. Second phase is the exponential phase. This represents the signal. In this phase, the fluorescent signal gradually increases, and a time reaches when it is exactly the double. Third and the last phase is the plateau phase reaction, and it denotes it may be due to the exhaustion of the DNA primers or exhaustion of the nucleotides in the reaction mixture. Threshold value is critically important and is determined experimentally. It is just a value which is of statistical significance. Now, the important point, CT value. The CT value is just the number of cycles at which the amplification curve passes the threshold. For example, in case of COVID-19, the CT value is generally in between 20 to 30, which denotes that the patient is positive. Now, since we know the basics of PCR, let's deep dive into the real-time PCR. But in first place, why do we need it at all? As we all know, PCR was invented in 1983 by Carey Mullis. At that time, PCR was not as simple as it is today. In fact, the earlier versions of PCR lacked automations completely and used three separate water baths for maintaining different temperatures. Enzymes, since were not heat stable, needed to be added manually during each cycle, resulting into an increased risk of contamination, high cost, and labor. Furthermore, the only way to know if the PCR worked properly was techniques like gel electrophoresis, HPLC, and DNA hybridization. The use of mineral oil overlay or paraffin wax as vapor barrier further added to the complications of the downstreaming processes. These all factors resulted in a necessity to create a technique which would eliminate the downstreaming process and detect the amplified DNA simultaneously without opening the reaction mixture at all. Now let's study about the real-time PCR itself. Here, the authors first performed the experiment to amplify the human HLA-DQ gene. They used samples. They used samples of patients. They used samples of humans and primers specific to the HLA-DQ genes. Here, they incubated ETBR in an increasing concentration and ran it for 20 cycles in a PCR machine. The amplified products were run into a gel electrophoresis, which showed the presence of expected product at various ETBR concentration. Ethidium bromide, owing to its hydrophobic ring, easily intercalates between the bases in a double-stranded DNA and showed the fluorescence when exposed to UV light. This experiment is important as it proved that the presence of ETBR has no obvious effect on the yield or specificity of the amplification and does not inhibit the PCR reaction. Second, this experiment was performed to detect the human Y chromosome. Here, the authors took the samples from male and also took the samples of female, which they took it for the purpose of a negative control. They ran it into a PCR machine, and the fluorescence was measured later. As expected, the samples containing female DNA did not show any fluorescence at all due to the absence of Y chromosome. The amplification curve created here, as we discussed earlier, formed a sigmoid shape. The experiment was successful and a threefold increase in the amplified DNA was observed. This experiment was important as it proved that more the sample of DNA at the starting of the experiment, the fewer cycles were needed to give a detectable increase in the fluorescence and hence a lower CT value. Third, the detection of allele-specific human beta globin gene. This experiment was solely performed in order to demonstrate the use of real-time PCR in genetic screening and detection of sickle cell anemia mutation. Here, they took samples from both homozygous and heterozygous patients containing the sickle cell anemia mutation. The tubes on the left side had the white primers containing the wild type allele specific for the mutation, and the right had primers specific to the sickle cell allele mutation. As it is shown, the difference was clearly seen in the fluorescence.
Chaitanya, can you wrap
Yeah.
up because the time is.
Yeah, sure. I'm ending it. Yeah. Lastly, we shall discuss the applications of real-time PCR. The applications of real-time PCR is great and enormous, so much that it could not be mentioned in just a single slide. As we all know, the real-time PCR has been used in grave times, such as in case of COVID-19, detection of cancer, genotyping, trisomy cases, etc. At last, the PCR field is ever increasing, continuously evolving, which has resulted into 21 different variants of the PCR. I'm sure that Carrie Mullis would be super proud and delighted when looking down at how magnificently his idea, which was perceived when driving a car, has developed into a technique which has been helping mankind and driving them towards a prosperous future. Thank you. Thank you for patiently.
Hello, Chaitanya, can you hear me?
Yes, ma'am. Sure.
Yeah, it was a well investigated and a well presented paper. Good one. Okay, I'm sure you all you have mentioned about the application of RT-PCR in COVID times, right? You have specified the same, if I'm not wrong. Okay. So I think everything is justified. That's all. Best wishes. Okay.
Yeah, thank you, ma'am.
Yeah, no question from my side.
Yeah. So.
Rachna ma'am, we can
Yes.
have the next part.
Yeah. I'll just announce the next person. Now, next to present is Icha from FY, and the next is Ketki Rane from Part 2. So I will make Icha the co-host now. Ketki Rane, you be prepared for the next presentation. Icha, you can start.
Ma'am, can you make me the co-host?
Yes, sir. No need.
Is the screen visible?
Yes.
Good afternoon, one and all present here. I'm Icha Shah from FY Biotech, and today I'll be presenting a paper based on replication of DNA in Escherichia coli by Matthew Massensen and Franklin Starr, which was communicated by Max Delbruck in May 14, 1958. I would like to share a little backstory on how Massensen and Starr met. Matthew was a lucky student to become Linus Pauline's, a chemist, last graduate student. He enrolled in his freshman chemistry course. The only way he could enter biology was through molecular biology. Apart from Linus Pauling, another person who saw biology from that point of view was Max Delbruck. He asked Matthew to read a paper presented by Watson and Crick. A few months later, Watson was going to teach physiology at Caltech. That's where Matthew and Frank met, and they found out that they had deep common interests. A year later, they decided upon testing Watson and Crick's hypothesis, which was the foundation of their quest. Watson and Crick had presented a model of double helix DNA structure. Crick's idea was about how the base pair were linked on the chains, whereas Watson's idea was about how the base pair was structured by complementary base pairing. From this model, they proposed a hypothesis in which the DNA replicated. It involved two chains coming apart, one acting as a template for synthesis of a new chain on its surface. This was a semi-conservative model that they had put forward. However, Max Delbruck, on the other hand, had opposed to this model. He put forward a model of DNA replication that followed a dispersive model hypothesis, in which breaks are produced in the parent molecule and it replicates and then carefully sealed up in a certain way. Whereas there were few other scientists who proposed a conservative model of replication. So now there were three targets out there that in principle could be distinguished if we could trace the fate of the old chain. Matthew's idea from the very beginning was somehow using stable isotopes to impart upon a DNA a different density. They first grew E. coli in a medium containing heavy isotope of nitrogen, N15 ammonium chloride, for number of generations. They next resuspended the cells in a medium that had ordinary nitrogen, which was N14 ammonium chloride. Later, a sensitive method for separating DNA according to its density was devised. The method they used was equilibrium density gradient centrifugation in cesium chloride gradient to measure the molecular weight. The process was based on sedimentation and diffusion, wherein the DNA molecules were driven by centrifugal force into a region of equilibrium where the solution's density equals buoyant density. This means if the DNA was heavy one, it would tend to be down near the bottom of the tube where cesium was concentrated and density was high. And if the DNA was a light one, as in having N40, it would be higher up to the tube. This led to band formation, and the photographs were taken during centrifugation cycle using UV absorption. You can see that as the number of cycles increased, the amount of N14, that is light DNA, increased exponentially with the time. The band in between N15 and N14 is the intermediate band, and it has density halfway between light and the heavy DNA. The results obtained from the experiment supported the hypothesis of semi-conservative model of DNA replication, which was proposed by Watson and Crick. They devised three conclusions from this experiment. First, nitrogen in each DNA molecule divided evenly between two subunits, which challenged Debrett's dispersive model. Second, each new DNA double helix contained one parental subunit, which supported the semi-conservative replication. And third, for every parental DNA molecule, two new molecules were made. Therefore, the amount of DNA after replication increased by a factor of 2. Matthew and Starr revolutionized genetics. This enabled the researchers to explain how DNA replicates, thereby providing a physical basis for genetic phenomena of heredity and diseases. It could be understood that semi-conservative replication provides DNA many advantages. It is fast, it is accurate, and it allows easy repair of DNA. It is also responsible for phenotypic diversity in few prokaryotic species. Lastly, but not the least, the experiment forms the basis of PCR technique, which was used in genetic testing, forensic, paternity testing. This experiment opened many doors for future explanation. We can now explain differentiation of cell from single cell into different types, protein synthesis, etc., which can now be used to study behavior of a mutant cell like cancer. Thank you.
Yeah, Icha, good one. Very neatly presented. Okay, but I just want to know what made you select this particular paper. Any specific reason for the same?
No, ma'am, I was searching for multiple papers and this was the first one that showed up.
Okay, fine. Yeah, it's one of the popular papers, but I just wanted to know if there was any particular reason as to why you have gone for the same. Okay. Anyway, good luck. Thank you.
Yes. Yeah, nice presentation. No questions.
Okay, so the next presenter is Kriti Shukla.
Rachita madam, just request
Sorry, sir.
that Excel sheet which you want me to enter the marks. I'm
Yes.
not getting any what you say permission for that.
I'll give
I have already sent the request, but I'm not able to fill in that.
it. You want me to send it to RJ College?
Uh, Actually, I've already received, but they said ki I'm not having edit access. So I have
You
already
already at
requested. I have already requested, but still I'm not able to do the
just check
edition.
if there is any.
Yeah. Yeah. Otherwise, it's okay. I'm writing in the another Excel sheet, and later on, then I will share.
You are just able to view but not able to check.
Yes, yes, yes. Let it be. I'll do that later on. Let the next speaker come.
So the next student is Hitki Rane.
Yes, ma'am.
The one after that is Kanaya Sen. So be ready, Kanaya.
Ma'am, shall I start?
Yes.
Okay. Good afternoon, respected judges. I'm Ketki Rane. Today I'll be presenting the paper on DNA sequencing with chain terminating inhibitors, which was published in the Proceedings of National Academy of Sciences Journal in December 1977. In this particular paper, the authors have described how the DNA sequencing can be done using certain chain terminating inhibitors. And this particular method is also commonly or famously known as Sanger's method of DNA sequencing. So in this particular method, as we know, for the DNA replication or DNA polymerization, we require the DNTPs, that is deoxyribose nucleotide triphosphate. Here on the 3' position, there is this hydroxy group, OH group, which is attached, and to this OH group, as we know, the phosphate group of the subsequent nucleotide gets attached, and further the DNA polymerization takes place. But here the authors had proposed that if they use certain chain terminating inhibitors like the DDNTPs and the arabinonucleoside, the termination of the DNA sequence can happen, and thus we can know the nucleotides which are present in the particular DNA. So in the DDNTPs, as we can see on the 3' position, the OH group is missing, because of which the phosphate group of the subsequent nucleotide won't be able to get attached over here, and the DNA chain will get terminated, because of which the DDNTP which got incorporated can help us to determine which is the nucleotide that is there in the particular DNA sequence. So they had used these two chain terminators for sequencing the DNA. Now in this diagram, as we can see, if the DDGTP gets incorporated, then the chain will get terminated at G, and we can know the first nucleotide is G. Next, if the DDTTP gets incorporated, then the chain gets terminated at the nucleotide T, and hence we can know the second nucleotide is T. Likewise, the entire DNA can be sequenced. So for this particular sequencing, the authors had used the DNA from the bacteriophage 5X174, and they had carried out this experiment. So for the experiment, they had used the 5 tubes. In each of the tube, the buffer, the sample, that is the DNA template, DNA polymerase was added, and the 4 DNTPs were added. And in each of the tube, they had added the chain terminators. So DDTTP, DDATP, DDGTP, DDCTP, and the arabinosyl CTP was added. So after this, for the annealing to happen between the template and the primer, they had used the Mn buffer. Now, why Mn buffer was used? Because it contains magnesium chloride, and magnesium chloride makes it difficult for the DNA polymerase to distinguish between DDNTP and the DDT, to distinguish between the DNTP and the DDNTP, so that the chain terminator DDNTP can also get incorporated into the sequence, and hence Mn buffer was used. And further, the incubation was done at 37 degrees Celsius for 60 minutes. After the incubation, the acrylamide gel electrophoresis was carried out. Now, this is the result where the primers A14 and A12D were used. Using these primers, this result was obtained. Here, the each of these alphabets indicate the chain terminators which are used. So, G indicates for DDGTP, A for DDATP, and likewise. After the autoradiography, the bands of the DNA were obtained, and the sequence of the DNA here they had written, we have to read it from left to right. So as first sequence over first nucleotide here is C, so the band was observed in the C lane, and hence the first nucleotide which we can predict is C. Then the second nucleotide which was observed immediate above this first C band was again in the C lane, and hence the second nucleotide is C. Likewise, the entire sequence of the DNA can be predicted. This is the result where they had used the primer R4 fragment, and the electrophoresis was carried out, and the sequence of the DNA was found out. And here, this is the result where the primer fragment A8 was used and the sequencing was done. Now, the advantages of this particular method is it has many advantages over the plus minus method of DNA sequencing, which was also described by Sanger in 1975. Here, there is no preliminary extension that is required. Here, we only require the DNA polymerase 1, and the sequencing can be done. Also, clear cut bands were observed, so that is also one of the advantage. And the the radio labeled triphosphates which are we use, that is DDNTPs which are used, they are labeled with the 32 phosphorus, which is a radioisotope of phosphorus. So these were the various advantages. And the difficulties faced by the authors were that the piling of the bands, that is the bands overlap with each other. So to understand which is the exact nucleotide, that becomes difficult. Hence, one has to not completely rely on the results which are obtained by the Sanger's method of sequencing. The confirmation should be done through some other method, and also we can do it by priming on the opposite strand. Another one important difficulty which the authors had faced was to obtain the DDGTP, because it was not commercially available that time in 1977. So they had to synthesize it. Also, this entire process of synthesizing the DDNTPs is quite expensive. And hence, these were the various difficulties that were faced by the authors. But this particular method of Sanger sequencing provided the basis for various sequencing methods that we have today, because of which the sequencing of DNA has become very easier nowadays. This is the reference. Thank you for the patient hearing.
Yeah, Ketki, you are in MSc, right? You have done it very nicely. You know, you have mentioned about the Sanger's method. You might have also read through the Maxim and Gilbert method, if I'm not mistaken. Have you?
Yes, ma'am.
Okay. And of these two methods, you know, which one has got a better DNA sequencing efficiency? Do you have any updates on that?
Ma'am, actually, the Maxim Gilbert method has more efficiency. because there we use the certain chemicals, and it is based on the chemical termination method, uh, which will cleave the DNA at particular sites.
Okay. Okay.
Mhm.
Yeah, fine. Thank you. That's it.
Thank you, ma'am.
Yes, thank you.
Sir, you want me to share this uh, Excel sheet in Gmail of
I have already received that in Gmail.
in
My
RJ
yeah,
at
yeah,
RJ College ED, no?
yeah, yeah. But they are asking me to request for edit access, which I've already said.
Yeah, so I can
I've
see
sent the
that, but I'm
message.
not able to give it.
Okay, fine.
Don't know
No, no
something is.
problem. I have another sheet wherein I am just putting my marks, and then later on I'll send it to you. No problem.
Okay, okay, sir. So next is Tanaya. I hope everyone has filled the attendance form, which has been shared in the chat box. Students, those who have not filled the form, please fill it up for our record.
Can I begin?
Yes.
Yes, yes, you can start.
Hello everyone and good afternoon to all. My name is Sonoya Sen and I'm a second year student of biotechnology. Today I would be talking on a classical paper with the topic acrylamide gel as a supporting medium for zone electrophoresis, which is written by Samuel Raymond and Louis Weintraub and published on May 2nd of 1959. Electrophoresis is a laboratory technique which was used to separate the DNA, the RNA, or the protein molecules based on their size and electrical charge. An electrical current is used to move these molecules on a gel. The pores in the gel work like a sieve, which allows the smaller molecules to move faster and the longer molecules to stay near the wet region. The abstract of this paper is the acrylamide polymerized in buffer solution forms a stable, flexible, transparent gel, which is useful in zone electrophoresis. You may be asking, why was this paper proposed? Since we all know that many different forms of stabilizing media have been proposed to the use in zone electrophoresis. Smith has described a starch gel technique. The use of gelling agents to stabilize a migration medium has been receiving an increasing attention. This paper describes a new commercially available synthetic gelling agent, which has many advantages over previously described agents for electrophoresis. Cyanogen 41 is a product of American Cyanamide Cooperative, which is a mixture of two organic monomers, that is, the acrylamide and N1-methylene bisacrylamide, in proportion which helps in producing stiff gels from dilute aqueous solution when it has been properly catalyzed. The process by which the gels are formed is a polymerization cross-linking reaction. These gels can be formed with various buffers according to the direction being supplied with the material. The precaution must be observed of covering the surface of the solution so as to exclude the oxygen when casting thin sheets or films of gel. This is easily accomplished by floating a sheet of non-wettable plastic on the surface of the monomer solution during the polymerization period. The contents present. What is found to be is that 3 to 5% of cyanogen in acid or alkaline buffer produces a satisfactory gel for the use in electrophoresis. The gel is optically clear and colorless. The optical density of 5% gel is 1 mm thick. The gel is flexible, elastic, stable, and completely insoluble in water once it has been formed. At a concentration of about 5% and more, it is strong enough to support its own weight, but it is somewhat brittle unless a plasticizer such as glycerol is included in the formation. This is how the gel appears to be looking like. Done. The rate of migration of serum albumin in 0.1 molar tris buffer with a pH of 9, gel with 5% cyanogam is approximately having a field of strength of 1.2 per centimeter per hour when a 10 volt per centimeter is being applied. The serum pattern can be developed in this medium in 8 hours when fixed in dilute acetic acid and stained with bromophenol blue as in any of the current standard procedures. Washing of this dilute acetic acid turns the gel a pale yellow and leaves the protein pattern dark blue as shown in this image. The pattern observed is sharper than in any commonly used medium. Way of preserving the pattern, whether stained or not, is to allow the gel to dry out completely. In the drying, the gel strings uniformly in all dimensions, producing a thin, flexible, transparent celluloid-like film which preserves the original pattern. This film later can also be rehydrated to its original form or dimensions by soaking in water. These are some of the images showing the drying process. Applications of electrophoresis is being used in forensic sciences, conservation of biology, and medicine. It is also used in DNA segments or for DNA fingerprinting to investigate the crime scenes, to analyze the results of polymerase chain reaction, in DNA profiling for taxonomic studies. This is how the acrylamide gel, which we all know about, is being used for electrophoresis first came into picture. Thank you everyone for being such a wonderful audience.
Yeah, Tanaya, it is a good presentation. I would just wanted to ask one simple question, like why you have chosen this particular topic? Any specific reason?
So I was finding about electrophoresis and I found this very interesting as we never knew how an acrylamide gel came into picture, how it was being used. So I really like this topic.
Okay, yeah, thank you.
Tanaya, I had logged out. I couldn't get the session. Do not worry. I'll go through the recording and I'll rate the things accordingly. Okay. Very best wishes.
Okay, ma'am, thank you.
So the next presenter is Lalit Wag.
Yes, yes, ma'am.
Yeah, yes.
Ma'am, is the screen visible?
Yes, yes, it's
Yes.
visible.
Okay.
You
So,
can
a very
start.
good afternoon. Okay, thank you. A very good afternoon to respected judges and all the participants present here. Myself, Lalit Anil Wag. I'm pursuing my master's degree in biotechnology and currently I'm in the second year. So today in this Anusandhan 2022, I'll be presenting a classic paper named Identification of a Novel Cell Type in Peripheral Lymphoid Organs of a Mice. This paper was published by Ralph Steinman and Hanvil Cohen around January 1973 in the Journal of Experimental Medicine, volume 137. So when the scientists were working on this mouse spleen cell, they saw that some of the cells were adhering to the glass and they had some distinct properties or morphology than usual lymphocytes. Then this made these two scientists to ponder upon what are these heterogeneous cell population, and this was how this paper was come into place. So here was a simple protocol wherein these specific strains of mice were used. Then the spleen was extracted, and then spleen adherent cells were prepared using enzymatic preparations of physical disaggregation. and only those cell suspension which were adherent were selected for further analysis. The cells were examined using phase contrast microscopy, wherein live cells were able to be uh, screened for, and then electron microscopy was used. Even staining procedures were carried out to which would help us to reveal the interiors of these cells. So what results did they get? Let us just look into it. First, the phase contrast observation, wherein we all know how these granulocytes, lymphocytes, phagocytes all look. But this four different variety of this cell had a bit distinct morphology. They had these varying length, width, forms, and shapes. Had pseudopods, which were very long, but had blunt terminations. Even the cytoplasm were dense with granules. The nucleus was large, contorted in shape, and were refractile. The nuclear membrane was again thick but uniform. The nucleoplasm also had this dense chromatin granules and all. And yes, this was something that they were able to observe in this phase contrast observation. Then by vital staining, they were able to see that the cytoplasm of such cell, which were quite distinct from the usual ones, had some lysosomes, some perinuclear granules, even some nuclear bodies. That not only one, 2 to 4 nuclear bodies, which were been seen after staining with these basic dyes, which were being used for the experimentation. Then further micro cinematographs of phase contrast revealed the movement pattern of these cells, and it showed that how unusual shapes were being able to be seen when they were able to go through this micro cinematographs, and with the help of pseudopods these cells were able to move also. Then after phase contrast, electron microscopy was done. Means we had also completed with staining and all, and now electron microscopy was done. This revealed that these elongated cells had relatively very smooth surface, contained large mitochondria, even rough endoplasmic reticulum. Some membrane-bound lipid droplets were also present. Here, the comparison reveals that these cells, which were unique, which were not named till now, are quite complex in structure and than these simple lymphocytes. They had these Golgi apparatus, mitochondria, so many not things which were present, which were not comparatively seen in these simple lymphocytes. Even the perinuclear region of these dendrite cells, which were not known till now, contained all the above such granules which I had mentioned, organelles which I mentioned. Even the microfilaments of these cells were around 80 angstrom in length. The ribosomes were relatively few and they were like scattered forms, forming polysomes. Then we did this tissue distribution of the cells, where these cells were present, and the table is in front of us. This revealed that spleen contained approximately 1 to 1.6% of these cells. Even mesenteric node, axillary cervical nodes, even Peyer's patches had these cells present in them. But these cells were absent in thymus, and but thymus contains some similar like cells which had these macrophage like functions and lacked any immunoglobulin receptors, but they were not these cells which were identified. There were some different cells. So we'll be seeing that in discussions. So yes, these all properties summarize that a novel type of adherent cell was found, which was more prevalent in spleen cells. They were large, contented refractile nucleus, cytoplasmic processes, as I said. Even they were present in Peyer's patches, the lymph node, the spleen, the bone marrow, but absent in thymus, intestines, and peritoneal cavity. Both nucleus and cytoplasm were weakly stained with basic dyes, and ruffling was seen. Abundant lysosomes and phagocytosis objects were there. In the living state, in vitro, its cytoplasmic processions are continuously elongating and retracting and reorienting themselves, leading to various different sorts of shapes. As I said, these cells had some similarities, which were not like had these macrophage function. They were also called cells, which were called dendritic cells, simply because of the processions that they were free. They had these fibroblast-like cells contained moderate amount of RERs, some cytoplasmic filaments and all. and they retained their some macrophage wala functions, having similar morphology, but they were quite different from it. Even primitive reticular cells were having some similar morphology, but they were not the cells which we were interested, or which the scientists were interested into. So in a whole packet, what we can say, a different type of cell which had distinct morphology, some extra added organelles, some dense structures in it, with a nice smooth surface and all. So we called this novel cell as a dendritic cell. And yes, this is how this dendritic cell was discovered by Ralph Steelman and Cohen. So these were the references they had used that time. And yes, thank you, ma'am. Thank you, sir.
Very nice, Lalit. That was a very illustrious kind of a presentation. I just have a query towards the end, all these evidences, either cytological or electron microscopy, phase contrast, all these are helping in the identification of the structure. Okay. And your last slide,
Yes,
is
ma'am.
mentioning about the, this one, correct. It's talking about the functions, the fibroblast like cells appear so and so, and they the functions, morphological functions. I'm just trying to figure out, was there any particular technique for understanding the functional aspects of this cell?
Ma'am, in the paper it is not mentioned, but
Okay.
I'm assuming
Yeah.
that they might be just how phase contrast that cinematograph pictures they were able to examine.
Correct.
So based on that, they might be able to figure out that they were showing such macrophage wala functions.
Okay.
So based on those observations, they might be predicting it.
Correct.
Not accurate predictions, but predictions.
Yeah.
Simple.
Okay, fine. Yeah. Thank you.
Yes.
That's it.
Yes.
And later, using these techniques, they could have introduced some pathogens in
So
a
as to
similar phase
understand,
contrast manner.
so as to understand whether phagocytosis is happening or
Yes.
not. Yeah. Okay.
Yes, good presentation.
Thank you, sir.
Next is Vaishnavi Menon.
Yes, ma'am.
After Vaishnavi, Kirti will be present. Kirti, are you there here? Kirti is not there. Next will be Dhanashri Parab.
Ma'am, shall I start?
Yes, you can start.
Good afternoon, everyone. Myself, Vaishnavi Menon, and today I'll be speaking on paper written by Nobumichi Hozumi and Suzume Tonegawa in the year 1976. The title of the paper is Evidence for Somatic Rearrangement of Immunoglobulin Genes Coding for Variable and Constant Regions. This is the front page of the paper that I have selected. The most likely explanation for finding of a single constant region amino acid sequence arranged in tandem with different variable regions was therefore that a single constant region gene segment was cooperating with multiple variable region gene segments in different cells or situations to generate the light chain gene. However, this notion was heretical to a generation of scientists brought up on the concept that one gene encodes one polypeptide chain. Now, this theory means that each gene is responsible for a synthesis of single polypeptide, and many experiments were carried out to prove this hypothesis. The experiment of Hozumi and Tonegawa was designed so as to determine whether the DNA encoding immunoglobulin light chain constant and variable regions existed in separate segments and in non-antibody producing cells, such that a single constant region gene could associate with different variable region genes in different B cells. Now, they hypothesized that This might be the case because of immunoglobulin amino acid sequencing data showing that the sequences of constant regions of many immunoglobulin light chains were identical. They reasoned that if multiple copies of a constant region gene existed, then each copy would be expected to accumulate silent or neutral mutations over time. Now, however, this experiment asked an entirely new question, that can a single piece of DNA change its place on a chromosome in a somatic cell? So to serve as their source of germline DNA, Hozumi and Tonegawa used DNA from an organ in which immunoglobulin genes would not be expressed. In this case, they used the embryonic liver. Now, for a B cell DNA, they use DNA from an antibody producing plasma cell tumor line, that is the MOPC321, which secretes the functional kappa light chains. They separately cut both the sets of DNA with the same restriction enzyme, and they use some radioactive probes to determine the sizes of the DNA fragments that they were using. So now let's see how Hozumi and Tonegawa conducted their experiment. They obtained a purified genomic DNA from the embryonic liver cells and from the MOPC321 tumor line. They then cut the two sets of genomic DNA with the restriction endonuclease BAM-H1 and separated the fragments using electrophoresis. Now they made 125 labeled nucleic acid probes specific to the two regions of the mouse kappa light chain. One of the Tonegawa's probes was a full-length radio-labeled piece of mRNA coding to the entire kappa chain sequence, and the other was 3' half of the sequence, which would hybridize to the constant but not to the variable region of the kappa chain gene. They then probed the nuclease-generated DNA fragments to determine the size of the fragments. Now, we would normally use a Southern blot procedure for electrophoresing the fragments, blotting the gel with the nitrocellulose paper, and then probing the paper with enzyme-labeled fragments. But Hozumi and Tonegawa's approach was a little more time-consuming. They cut the gel into 30 slices, melted the agarose, and then separately eluted the DNA from each slice. Now, to each DNA sample, they added the radio-labeled RNA and allowed it to anneal. Now, after annealing, they removed the unannealed RNA by using RNAs. The radioactivity remaining in each fraction was then plotted against the size of the DNA in the slice. In embryonic liver, the DNA sequences encoding the variable and constant regions respectively were located on different restriction endonuclease fragments. However, these two sequences were co-located on a single restriction fragment in the myeloma DNA. The kappa light chain was formed by DNA recombination between the variable and constant region gene segments. Now, the original paper speaks of purifying their own restriction endonuclease, that is BAM-H1, from a bacterial sample which they collected from one of their colleagues and performing the electrophoresis at 4 degrees Celsius for 3 days. Now, they therefore probed each sample with 3' constant region probe, as well as the one that bound to the whole light chain, and then inferred 5' variable region by binding by subtraction. So these are the suggested this plot shows the amount of radioactivity in each fraction. This reflects the molecular weight of the DNA fragment. Hybridization patterns with the DNA of two different sources, that is the embryo and MOPC321 tumor, were superimposed. Now, these are the suggested models of the variable chain and variable and constant gene joining at the DNA level. In the copy insertion model, a specific V gene is copied and duplicated and inserted in a position near the constant gene. In the excision insertion model, the V gene is excised or removed by forming a episome-like structure, which we can see here. and this is integrated to the adjacent constant chain gene. In the deletion model, the interval between variable gene and the constant gene loops out and it gets deleted. In the inversion model, variable genes and the constant genes uh, are arranged in opposite directions. After the arrangement, this segment gets inverted, the V region being the inclusive part, that is which gets included, and the C region, that is the constant gene region, being the exclusive part, that is it does not get included in the uh, inversion. Now, how did this paper affect what we know now? Now, gene rearrangements were unheard in mammalian DNA. This field exploded in the last 15 years. Soon thereafter, the V gene was divided into three segments, that is, the variable, diversity, and joining. And all these had to be rearranged too. The studies of mechanism of rearrangement uh, led to the discovery of hepta and nonamer DNA sequences, and finally to the elusive RAGs themselves. Now, during ontogeny, it was found that the V genes were arranged in linear order, and this led to the mapping of genes along the chromosome and chromatin accessibility model. The basis of allelic exclusion was elucidated, plus all these mechanisms then applied to rearrangement of the newly discovered TCR genes, that is the T cell receptor genes. The impact of this experiment on the biological community was profound, and for the first time, DNA was shown to be cut and rejoined during the process of differentiation. Thank you.
Yeah, Vaishnavi, well presented, and all illustrations, and you have covered up with the methodology and everything. Very nice. Okay, no questions.
Thank you, ma'am. Ma'am, should I stop share?
Yeah, you may. Anil sir, your comments, please.
Uh, yeah, definitely well presented. I really do not have any questions. Best wishes.
Thank you, sir.
Yeah, good Vaishnavi. Now we go to the next student. It is Dhanashri Parab.
Am I audible, ma'am?
Yes.
I can share the screen.
You're audible.
A very warm welcome to Anusandhan 2022 on the occasion of National Science Day. Myself, Tanashri Gajanantara from first year of Biotechnology Department. Today, I did like to take this opportunity to share with you a classic research paper titled as The Studies on the Chemical Nature of the Substance Inducing Transformation of Pneumococcal Types, in, which includes induction of transformation by a desoxyribonucleic acid fraction isolated from pneumococcal type 3, which was published
Your
on
screen
1st
is
of...
not visible, Dhanashri. Have you shared the presentation?
Yes, ma'am.
Please, can you share it again because we can't see. Can
Yeah,
you
it's
see,
not
sir?
visible. No, it's not visible.
Yeah.
Yeah, we shared. Just 1 minute. Now is it visible, ma'am?
Yes, now it has started. Yes. Yes. Start.
Yes. Can I continue
Yes.
or should I start from beginning?
No, it's all right.
Yeah. So I will start with the um, yes. So this research was done by three great scientists in the Hospital of Rockefeller Institute for Medical Research, named as Oswald Oswald Every, Colin MacLeod, and Maclean McCarthy. Now let's take a look at this scientist and go further. Going ahead in detail about the paper. Now the present paper is concerned with a more detailed analysis of phenomenon of transformation of specific types of pneumococcus. The major interests are centered in the attempts to isolate the active principle from crude bacterial extracts and to identify its possible chemical nature, or at least to categorize its sufficiency to place it in a general group of known chemical substances. So, for the purpose of study, the typical example of transformation chosen as a working model was the one which they had most experience with and which seemed best suited for the analysis. Now, this paper represents the transformation of non-encapsulated R variants of pneumococcus type 2 to type 3. Knowing further about the transformation, British bacteriologist Frederick Griffith was the first one to suggest that bacteria can transfer genetic information, and he called that process as transformation. Then further, it was researched by Oswald, Avery, MacLeod, and McCarthy. They came into picture and they attempted to determine the chemical nature of the substance that allows transformation. Then they reported that the transforming substance allowing transformation is the genetic material of the cell known as DNA. Now, further taking a look at the Griffith experiment, they are very closely related. So, I will describe in short what Griffith experiment was about. So, the Griffith experiment was a stepping stone for the discovery of genetic material. With Streptococcus pneumonia, we showed two patterns of growth. One cultural plate consisted of smooth, shiny colonies, while other consisted of rough colonies. The difference was due to the presence of mucus port in S strain bacteria, whereas the R strain bacteria lacked them. Now, Griffith injected both S and R strains to the mice. The one which was infected with the S strain developed pneumonia and died, while that infected with R strain stayed alive, as shown in the figure. Now, what he did in the second phase was Griffith he killed the S strain bacteria and injected into the mice, but the mice stayed alive. Then he mixed the heat killed S and live R strain. This mixture was injected into the mice, and the mice died. Then, in addition, what he found was the living Ashton bacteria in the dead mice. So, based on this observation, research concluded that the R strain bacteria had been transformed into S strain bacteria. The R strain inhibited uh, some transforming principle from the heat killed S strain bacteria, which made them virulent. And he assumed that this transforming principle is the genetic material that is known as DNA. Moving further, with the experiment. This experiment had some components included in the system, which were nutrient broth, serum, then strain of R. pneumococcus, and the procedure, which included extraction, purification, and the determination of the chemical nature for this principle. Now, for the experiment, they had some preparative methods. So that included to bring out the source material, extraction of histone cells, then they deproteinized and removed the capsular polysaccharide, then they did alcohol fractionation, and then after that they had tested for temperature and pH. Now I will describe the summary in short for the paper. The transformation of unencapsulated R variants of pneumococcal type 2 into fully encapsulated cells under appropriate conditions were made. Then different methods for isolation and purification were carried out and evaluated. Then what they did was they had to check for the genetic material, what intakes that transformation process. So they had to perform various chemical, enzymatic, and serological tests. Then they expressed those results in detail, and then they had to present the evidence that showing that chemically induced alterations in the cell structure and function, and then the hypothesis was reviewed. The conclusion of the paper was given as the evidence presented supports belief that a nucleic acid of desoxyribose type is the fundamental unit of transforming principle of pneumococcus type 3. Now I will let you know the significance of this experiment, which was very helpful for the entire world to carry out further research. So the first one was to make multiple copies of DNA or DNA cloning, which involves the insertion of a DNA sequence of interest into a vector by the process of transformation. The second one that I would like to list is to make large amount of specific human proteins, where the recombinant protein expression in the bacteria requires the insertion of a DNA fragment into an expression vector and then are transferred into bacteria through the transformation process, which very well helps to produce human insulin, which is nowadays used to treat type 1 diabetes and cure that. Further, it was used to genetically modify a bacterium or other cell. The bacteria is induced with the genes of the required modification by transformation, and that can be used for research activities and other things. Moving further, I would like to list that the bacteria are used as means of storing and replicating plasmids. Now, plasmids can be used as vectors to carry foreign DNA into the cell. Now, E. coli is preferred host for the protein production. Also, it has uses in the pharmaceutical industry for the production of vaccines and medicines where similar processes carried out. Also, it has different aspects and it can be included in agricultural field as well, where genetically modified crops are modified with the help of agrobacterium that is induced by the process of transformation. So the entire uh, significance revolves around the fact that the bacterial transformation has been very well helpful in every aspect of today's life. Thank you so much.
Yeah, nice presentation, Dhanashri. It's one of the classic and what you can say most popular experiments amongst the students, right? So very
Yes.
nicely done. Thank you.
Thank you.
Anil sir.
No question, madam. You can have next.
Okay.
Yeah. So the next student is Isha.
Yes, ma'am.
Isha Shah from FY.
Yes, ma'am. Is the screen visible, ma'am?
Yes, you can start.
Good morning, good afternoon to everyone present here. My name is Krisha Shah from first year BSc Biotechnology, and I am grateful for this opportunity of taking part in Anusandhan 2022 Classic Paper Presentation Seminar on this occasion of National Science Day. So as you can see, my topic for the Classic Paper Presentation is Sex-Linked Inheritance in Drosophila by Thomas Hunt Morgan in 1910. First of all, we are going to see the organism that was used for this paper. It's Drosophila melanogaster, that is fruit fly, which is a common fly which is found near fruits. Fun fact about common fruit fly is that Drosophila melanogaster and humans have genes that are almost 60% similar to each other. Drosophila is also used for study of some serious neurodegenerative disorders, including Parkinson's and Alzheimer's. So this little creature is nothing but a miracle for us. Thomas Hunt Morgan was the first one to actually use the small fly as a genetic tool. Here we are going to see why Thomas Hunt Morgan chose fruit fly as his study organism. The first one is short generation time. That's about 2 to 3 weeks. Second, rearing Drosophila is very easy as they feed on fruits, hence the name fruit fly. Third one, less number of chromosomes. As we saw that it has only 4 pair of chromosomes, 3 out of which is autosomes and 1 is sex chromosome. It is very easy to visualize them microscopically. Fourth, sexes can be distinguished easily as male fruit flies are smaller in size as compared to that of female fruit flies. They also lay large number of organisms per mating. We are going to see about Thomas Hunt Morgan. He is known to be the father of experimental genetics. He and his group, which was called as Fly Group, made tremendous contribution in understanding the role of chromosomes and genes in inheritance. Thomas Hunt Morgan was awarded Nobel Prize in Physiology or Medicine in 1933 for discoveries that elucidated the role of chromosome in heredity. So how this experiment came about? One day, Thomas Hunt Morgan was going through his vials, and one of the vials contained red-eyed flies, vials that contained red-eyed flies. But one fly that Morgan found particularly had wide-eyed male fly. Now this is unique because the wide-colored eyed fly was considered to be special type of mutant by Thomas Hunt Morgan. It was because all the other flies were having red eyes. Here, the wild type, we consider it as normally found in nature, that is natural forms, while mutant type is the one which is occurred due to random mutations. So according to this experiment, the wild type here is red-eyed flies, while mutant type is white-eyed flies. So for this experiment, Thomas Hunt Morgan first crossed red-eyed female with white-eyed male, as a result of which the first generation came out to be all red-eyed fruit flies. It had both sexes, females and males. For the second generation, that is F2 generation, Morgan crossed the first generation flies, and as a result of which he obtained the classic Mendelian results, that is the phenotype is of 2:1:1 and the genotype is 3:1. So here we can,
the other way.
Yes ma'am. So here in the F2 generation, we can see that we have 2 red-eyed females, 1 red-eyed male, and 1 white-eyed male. These are the charts that were provided to explain it more clearly. So over here, as we can see that the parent generation was crossed. For one, we had taken red eye and that mutant wide eye male fly, as a result of which all the F1 progeny were found to be red-eyed fruit flies of both sexes, that is female and male. Then they were crossed together, and as a result of which we found the F2 generation to have the wide-eyed characteristic that was there in the F1 in the parental generation. So as a result of which we can say that the wide-eyed gene, which was present in parental generation, was passed down to the F2 generation via F1 generation. Or now we can also say that the wide-eyed characteristic that the male fly had in the parental generation was passed down to the F2 male via F1 female. That is, the characteristic was passed down from grandfather to grandson via daughter. This experiment is basically to show the sex-linked inheritance of the genes that the organism possesses. Over here, we have the genotype and phenotypic chart over here. And it is very rare for female flies to have white eyes because this is X-linked inheritance. And as female flies have 2 X chromosomes, this is the condition that is to be fulfilled to get a white-eyed female. As I said, that the genes expressing white eye was carried through generation in following fashion. That is, from parental male, it was transferred to F2 male via F1 female. This type of inheritance was known as criss-cross inheritance. Not only this experiment made Thomas Hunt Morgan confirm the theory of chromosomal inheritance. Here are the conclusions that were derived. Eye-colored genes here are sex-linked, which means characteristics traits linked with sex chromosomes. These results obtained by Thomas Hunt Morgan confirmed the chromosomal theory of inheritance. Thomas Hunt Morgan put forward the assumption that the gene for eye color in Drosophila melanogaster are present on X chromosome. Now, why it is important for us that we study this experiment? It is because a lot of diseases that occur in humans and animals are related to sex. For example, in humans, color blindness, hemophilia, congenital night blindness, etc., are some of the diseases that occur due to sex inheritance, sex-linked inheritance. Thank you very much.
Well done, Krisha. At FYBSc, you have done such a wonderful presentation, and I think you have a long way to go ahead as far as research is concerned. Okay, all the very best. I just wanted to update you the centimorgan units. Of course, Morgan has contributed a whole lot as far as Drosophila studies are concerned. There is a unit called centimorgan, if at all you might have come across. Okay, this was a unit, which was given in the name of Morgan. Okay, so, and this is basically applied for sex linkages. To find out linkages and mapping, they use the unit centimorgan, and that is after the name of Morgan. Okay, so that was just an information which I wanted to give it. Okay, well done.
Thank you so much, ma'am.
Yes, well done. At FI level, definitely you have done wonderful presentation.
Thank you so much, sir.
Yes, best wishes.
Thank you.
The next student who will be presenting is Nimisha Sharma from MSc Part 1. Kiran sir has joined the session. Welcome, sir.
Am I audible and is my screen visible?
Yes, yes.
Okay.
Yes.
Just put it on slide mode.
Yes, ma'am. Good afternoon, everyone. My name is Nimisha Sharma, and I'm from MSc Part 1, Department of Biotechnology. And today I will be talking on the paper, which is titled as Mice Deficient for P53 are developmentally normal but susceptible to spontaneous tumors. Here are the list of authors, and here is an image of the paper which I have referred to. So starting with the introduction of P53, P53 was first detected in rodent cells, which was transformed by the simian virus, which is SV40. And the gene that encodes for P53 is a tumor suppressor gene, but it may behave as an oncogene in some of the mutant forms. A wild type P53 inhibits the tumor formation, and if when a human tumor cells are transfected with wild type P53 gene, they grow at a reduced rate and they have decreased tumorigenicity, which indicates the suppression of the tumor. A loss of a tumor P53 function may lead to a malignant phenotype. Deletion, rearrangement, retrovirus insertional inactivation of P53 are observed in erythroleukemias found in mice. So mutation or allele loss of P53 have been associated with a variety of human organs. P53 mutations are associated with inherited cancer susceptibility and Li from any syndrome. So a point mutation was been observed in P53 gene, which was specially seen on exon number 7. So talking about the normal role of P53, it has a basic function in regulation of the cell cycle. P53, which has been phosphorylase by CD2K kinase, it has which is required in mitosis. P53 is partially regulated and it accumulates in our cytoplasm in the G1 phase, and then it is seen to migrate to nucleus in the beginning of S phase. So transformed cells that are transfected with wild type P53, they all arrest in the G1 phase. As P53 is implicated in cell cycle control, it might be important for the embryonic development. So it is expressed in many tissues in mouse, however, in very low levels, and it has a short lifespan of about 20 minutes. So to generate any animal model and to check for the development or tumor formation, homologous recombination in mice were carried out in the embryonic stem cells to form a null allele of the gene. So firstly, a chimeric mice were generated and they were mutated. P53 allele was established in mouse germline. Homozygous mice which were homozygous for mutated gene are also normal, but they are susceptible to spontaneous tumors before they reach of around 6 months of age. Usually they form lymphomas. They are seen to develop lymphomas and sarcomas. Heterozygote, these are nothing but a single wild type P53, which rarely develop tumors till around 9 months of age. So it was concluded that a normal P53 is not required for a mouse development, and a loss of P53 is enough for predisposing the cells to a tumorous cells. So P53 gene targeting. So this P53 gene was interrupted by a Pol2 neo expression cassette and some deletion and selection, and it was screened by the PCR methods. Hybridization method indicated the disruption, the confirmed disruption of the P53 gene, and recombination had taken place in 2 colonies of the embryonic stem cells. P53 deficient mice. One of the cell clones were used to generate chimeric mice by micro-injecting in a three and a half day old blastocyst, and then it was implanted in a pseudo-pregnant female. The offsprings were which were chimeric were indicated by the agouti pigmentation, as we can see towards the right side. And to access the role of P53 in development, there was a P53 heterozygotes were also intercrossed. So how did they confirm for the p53 null allele was a 106 base pair probe was made, which was specific to a region of the p53 gene, which was deleted in the mutated cells. When hybridization was carried out, homozygotes did not bind any DNA fragment, which confirmed that both the copies which of the highly conserved region of the p53 were deleted. Tumor susceptibility. Coming to the main aim, mice which were mutant for p53 were able to test, and because of that, the effect of loss of tumor suppressor could be studied. Heterozygotes were studied, homozygotes, and a little wild type mice were monitored. So checking about the wild type, so out of 95 animals till around 9 months of age, all of them failed to develop any tumor or any illness. In one of the chimeric of around 14 months of age, two different primary tumors were developed, osteogenic sarcoma and ovarian choriocarcinoma. In the second chimeric of around 14 months of age, a Leydig cell which of the testes was seen. Till 9 months of age, out of 96, two heterozygotes developed tumor, where first heterozygote developed an embryonal carcinoma at around 5 months of age, and in the second heterozygote, malignant lymphoma was developed at the 9 months of age. So in homozygotes, out of 35, 26 developed tumors at around 6 months of age, and some also before 10 weeks also. This occurrence, this rapidly increased till the 15th or the 25th week. 6 out of 35 homozygotes died because of some unknown reasons. So all these tumors were analyzed by histopathology. Multiple primary neoplasms were observed in 9 out of 26 tumors bearing the homozygote mice. Now, mostly all the tumors that were observed were malignant lymphomas, around 20 out of 26 animals, but there was a little frequency of sarcomas also. The lymphomas involved a variety of tissues such as thymus, heart, lungs, spleen, liver, kidney, brain. Sarcomas were found. So there were 7 of them with hemangiosarcomas, 3 with undifferentiated sarcomas, and 1 with had developed osteosarcoma. So according to all the experiments carried out, looking at the discussions, so after introduction and all the screening processes, the p53 gene was successfully disrupted and then transmitted to the germline. And these homozygous mites which were obtained were completely normal, but they were susceptible to tumors. And tumor development was seen in 75% of the mice at around 6 months of age. And a variety of tumors, as we had already seen, in different kind of tissues were seen, which indicates that p53 is involved in tumorigenesis in many tissues. And some deaths were also there taken place, which whose reasons were unknown. To address those, there were many examinations of the thymus, spleen carried out, which resulted in normal CD4 and CD8 levels. Antibody isotoping was carried out, but all of them were normal. So if there was any immune defects present, so they would contribute to the tumor, but it is unlikely for an immune deficiency to be the sole cause of cancer. So the basic conclusion of this is that p53 was responsible for inducing a tumor, and p53 models are been highly used right now for studying all these anything related to cancer. So p53 was proven to be involved in induction of a cancer. This proved to be very useful for marker and for diagnostics of cancer. And it is also now used for, it is one of the targets used for gene therapy. These are the references of the paper. Thank you.
Yeah, Nimisha, very well presented. Just a question mark as to, do I have a substitute for a mice model to study the p53? Any idea on that?
Yes, ma'am. We can study like other than the C57. Okay, you're talking about a substitute for a mice model.
Yeah.
Okay. Nowadays, ma'am, for cancer, all the researches are going on mice model only.
Correct.
We can use different species if we want, but the main model required for cancer studies is mice.
Okay. Okay. Fine. Thank you. Good. Well.
Yes, thank you. No questions.
Okay.
Yeah, I think that will be the last.
Madam, I think Nandini, she
But
has presented.
she's not there in the meeting, so I have marked her absent.
Okay, yeah.
So let's see if she's there. Nandini Vats? No, she's not there.
She
Yeah.
has not presented.
So this is the last presentation.
Yeah.
And
can I start?
Yes.
Good afternoon, faculty members and my dear friends. I am Riya Bhivankar from SY Biotechnology, and my topic of discussion is primer-directed enzymatic amplification of DNA with a thermostable DNA polymerase by Randall K. Saikai et al. Before going to the heart of the presentation, I would like to throw some light on PCR, that is polymerase chain reaction. Firstly, let's get acquainted with the term PCR. It is a method widely used to rapidly make millions to billions of copies from complete or partial copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large amount to study in detail. So when scientist Carrie Mullis was working on a research paper to identify nucleotide sequence, he made a serendipitous discovery of polymerase chain reaction. In this process, the requirements are the DNA sample, which is to be amplified, that is, to make more copies of the DNA sample, primers, which help in locating the target DNA, whereas polymerase attach itself and nucleotide, that is adenine, thymine, guanine, cytosine, which facilitate the extension of DNA fragment. And the main ingredient is the Taq polymerase, which helps in identifying the starting point from where the DNA synthesis process commences. And lastly, the buffers. All this is taken in a PCR tube, which is placed in a thermal cycler that provides appropriate environment for the reaction. Let us look at one of the PCR process. First is denaturation, that is, the double-stranded DNA is split into two single-stranded DNA. Next step is annealing, where the primers attach themselves to specific site of the DNA strand. And third step is the extension of DNA strands by addition of nucleotides. All of these reactions occur at varied temperatures. The role of thermostable DNA polymerase and the need for it is highlighted in the slide. Conventionally, the DNA polymerase from E. coli was used, the Klenov fragment, to catalyze the extension of DNA. However, this enzyme was unable to withstand higher temperature, which was required for separating the newly synthesized strands of DNA because of its thermolabile nature. Thus, fresh polymerase had to be added in each cycle, which was a tedious prone process and had many errors and could cause contamination. Hence, the alternative is a thermostable Taq DNA polymerase, which is derived from Thermus aquaticus. The advantage of Taq polymerase in PCR tests are that it can survive extended incubation at 95 degrees Celsius and is unaffected by heat denaturation. This is because it is a heat-resistant polymerase, which can substantially improve the overall performance of reaction. Now, we'll do a comparative analysis of Kleno and Taq DNA polymerase in PCR reaction. to see how the enzymes aid in the reaction. As observed in the first diagram denoting electrophoresis, where clenopolymerase was used from lane 1 to lane 5 and Taq DNA polymerase from lane 7 to 11. The results indicate that clenopolymerase showed non-specific annealing and extension of unwanted sequence, which was observed by improper band formation. Whereas it was observed that there was distinctive band formation with usage of Taq DNA polymerase. There are several factors that influence the specificity of DNA polymerase, catalyzed by amplification. A study was done firstly on extension time for primers, secondly on increasing concentration of enzyme, and third factor was by increasing the annealing temperature, as observed in the second diagram to the right of the screen. Figure 3 indicates successful amplification of beta globin gene because 9 out of 15 DNA copies were amplified, which a conventional enzyme would amplify successfully to a threshold limit of 4 copies. As seen in the diagram, which is appearing on the right to the screen, one can see a comparison of results of southern blotting using Cleno and Taq DNA polymerase. The Taq polymerase anneals the primer only to beta globin gene and thus is highly specific. On the other hand, clenopolymerase being unable to distinguish between beta and delta globin and amplifies both of them. Thus, it reduces the specificity of clenopolymerase. Advantages of using Taq DNA polymerase are many in numbers. Due to this advantage of Taq DNA polymerase, it has significant applications in PCR testing, like diagnosis of infectious disease or chromosomal aberrations, analysis of nucleotide sequence, and forensic analysis of genetic material. Thank you for your patient listening and being such a wonderful audience.
Thank you, Ria. I would like to thank the judges, Dr. Anil Awad sir and Dr. Bidu Achari for coming and judging our students and encouraging them by giving your inputs, valuable inputs. They would have definitely gathered a lot of knowledge from your inputs. Thank you very much. I will end the meeting now.